Answering MCAT questions in under 60 seconds. A newly characterized enzyme, HYDE1, catalyzes the hydrolysis of a series of structurally related substrates. Kinetic analysis shows that product formation is the rate-limiting step for all substrates tested. Which of the following conclusions is most consistent with stronger enzyme-substrate complex formation? Okay, so looking at these answer choices, A says substrate X shows a higher kcat than substrate Y. Well, what is kcat, right? Kcat is the turnover number, and that's given by Vmax divided by the total enzyme concentration. And this doesn't have anything to do with affinity. Rather, it reflects how efficient an enzyme is at catalyzing the reaction, the conversion of the substrate to the product at saturating levels of substrate when all the enzyme active sites are filled with substrate, right? It doesn't reflect anything about the affinity, so we can safely rule that out, okay? Because remember that a stronger enzyme-substrate complex formation is going to happen if you have a higher affinity of enzyme for substrate. Now B, substrate Z has a lower Km than substrate X. Well, that sounds correct, because remember that Km is inversely proportional to enzyme affinity, meaning that if you have a lower Km, you have a higher affinity of the enzyme for the substrate, okay? And so if you have a lower Km than substrate X, or Z has a lower Km than substrate X, then Z is going to be expected to form a stronger enzyme-substrate complex by virtue of that higher affinity, right? So this sounds correct. But let's rule out C and D. C says substrate Y reaches Vmax at a higher substrate concentration than substrate Z. Well, remember that the Km is also the substrate concentration at which the V naught, the initial rate, is equal to 1/2 the Vmax. So therefore, if Y is reaching Vmax at a higher concentration of substrate than Z, then that implies it has to have a higher Km, not a lower Km. And remember, higher Km means lower affinity for enzyme, right? So that's wrong. And then D, substrate X shows a steeper initial velocity curve than Z. Well, what is the slope, right, of the Michaelis-Menten graph, right? Well, the slope is actually going to be equal to what's called the catalytic efficiency, which is kcat divided by the Km. So it's this turnover number divided by the Km. And notice that the Km is present in that slope equation. However, we don't know whether the slope is greater because the kcat is higher or because the Km is lower, right? We don't have any information about that. For instance, substrate X and Z might have the same Km, or the enzyme might have the same Km with those substrates, but the kcat could be different. We don't know. So D doesn't really tell us anything specific about that. Okay, so we can rule that out. Whereas B is definitively suggesting Z is going to form a stronger enzyme-substrate complex.